This is a mole known as Avogadro's number 6.022 times 10 to the 23rd particles. A mole of water, a mole of methane, and a mole of nitrogen oxide contain the same amount of particles. Concentrations of elements and molecules are measured in moles per liter and denoted in brackets. These concentrations affect the chemistry within a cell. Each element and molecule possess a molecular weight determined by 1 mole of that element or molecule. Molecules and elements vibrate. There is an energy associated with the vibrations known as Gibbs free energy. The orientation and type of bond determine the chemical properties of molecules, such as water being polar and methane not. Amino acids are molecules, and molecules contain functional groups, such as amine and carboxyl groups. The amine group allows for amino acids to peptide bond, and the carboxyl group chemically acts as an acid. When amino acids bind to other amino acids through peptide bonds, water is released by using OH from the carboxyl group and H from the amine group. The difference in chemical properties of individual amino acids are determined by the R group. And the sequence of amino acids determine the function of the protein made from those amino acids. Pepsin is an enzyme, or protein, that is used in digestion, giving the body and cells the fuel needed to perform metabolism. Pepsin is made from a sequence of amino acids. The sequence of amino acids is determined by RNA. RNA is made of phosphate, phosphoric acid, ribose sugar, and nitrogenous bases: guanine, adenine, cytosine, thymine, and uracil. When bonds are formed between phosphate and ribose, another water molecule is released. When bonds are formed between ribose and a nitrogenous base, a water molecule is released. When ribonucleic acids polymerize and form bonds, another water molecule is released. Ribonucleic acid sequences of A, C, G, U are called RNA. Both RNA and proteins are sequences, one of ribonucleic acid and the other of amino acid. The sequence found in RNA determines its function, such as mRNA, tRNA, and ribosomes. The sequence found in proteins, amino acid chains, determines its function, such as enzyme, markers, or cytoskeleton. A ribosome is a sequence of ribonucleic acid that has become enzymatic, a ribozyme, and uses mRNA, a sequence of ribonucleic acid, and tRNA, a sequence of ribonucleic acid, to create a chain of amino acids. Different amino acids are attached to different tRNA, and these individual tRNA contain a different sequence of ribonucleic acid that interact with the mRNA template within the ribosome. There are 3 positions tRNA can take within the ribosome. In the middle position, the mRNA sequence CAU corresponds to a tRNA attached to valine, so only this kind of tRNA can bind here. This amino acid chain builds off this middle position. The right position will bind a tRNA with a sequence for GGC, and the amino acid attached to that tRNA is proline. Once the amino acids form peptide bonds, the ribosomes shift forward on the mRNA template, causing the left spot to release its tRNA and the right spot to become available for a new tRNA to bind. The tRNA that was in the right position becomes the middle position and holds the chain for the amino acids. GCU is the newest sequence on the mRNA and corresponds to a tRNA attached to arginine. The sequence of the mRNA determines a new amino acid every 3 base pairs until finally a protein is made, a sequence of amino acids. What is more interesting is a ribosome and its sequence of ribonucleic acid are put together by proteins. So a protein is made by RNA, ribosomes, mRNA, and tRNA, and RNA is made by protein known as enzymes. Amino acid and the nucleic acid concentrations affect each other. Phosphoric acid concentrations and the concentrations of its bases affect each other. Adenosine triphosphate concentrations affect adenosine diphosphate concentrations affect adenosine monophosphate concentrations. The same for the other nitrogenous bases. Don't forget thymine for DNA. Nucleic acid concentrations and phosphoric acid concentrations affect each other. The concentrations of enzymes like RNA polymerase and helicase affect the concentrations of DNA available to transcribe, affect RNA concentration, affect concentration of enzymes, affect concentrations of RNA, ribosomes, tRNA, mRNA affect concentrations of enzymes, affects concentrations of DNA available to transcribe through the concentration of enzymes that methylate DNA wrapped histones, where histones are protein, same as enzymes. So, RNA polymerase and DNA create a sequence of mRNA in the presence of concentrations of ribonucleic acid. The sequence of amino acids of the RNA polymerase enzyme allow it to facilitate the chemical reactions of bonding a new ribonucleic acid to the mRNA sequence. Likewise, enzymes are responsible for the chemical reactions needed to make ribosomes, tRNA, and mRNA. Enzymes may use energy sources like ATP and may physically change orientation to facilitate the orientation of molecules to catalyze a reaction to form a phosphodiester bond. The same is true that the sequence of DNA or RNA made from DNA or not makes the amino acid sequence that becomes the enzyme pepsin, which helps get the chemical energy to catalyze reactions from food, chemical energy used by helicase, an enzyme to unwind the DNA to allow RNA polymerase to bind to DNA and use chemical energy to catalyze the reaction of sequencing an RNA sequence that creates the ribosome. That catalyzes the reaction of sequencing an amino acid sequence that becomes the enzyme pepsin. And the same is true that mRNA, tRNA, and ribosomes create pepsin that helps get chemical energy from food, chemical energy used by RNA polymerase and helicase to create mRNA, sequence from DNA, or proteins creating proteins that manipulate DNA. To understand how enzymes, proteins catalyze reactions, a graph of the progression of a reaction and the energy of its reactants and products shows a curve. E and progress denoted T. A plus B is in equilibrium with C plus D. The energy of activation is the amount of energy needed to excite the vibrations of reactants to begin reacting. KQ is the relationship of the concentrations of products and reactants in equilibrium to determine rate of reaction. Catalysts and enzymes lower the energy of activation. Energy of activation affects how readily a reaction will proceed. Reversible reactions proceed forward and backwards in equilibrium, and the pH is determined by the concentration of hydrogen ions. If the concentration of hydrogen ions increases, for example, by adding an acid, the pH lowers. Acids and their conjugate bases are also in equilibrium with each other, as well as the concentrations of hydrogen ions. Cells are maintaining an equilibrium with the reversible reactions of anions, cations, elements, carbohydrate, amino and nucleic acids, and lipid concentrations. Even like chirality of molecules, acids, bases, and water, pH changes can affect enzymes and their efficiency. Adenosine triphosphate and the other nucleoside triphosphate are in a chemical dance with each other as RNA and DNA are constantly depleting and replenishing their concentration. At the same time, your body is synthesizing the proteins to combine their precursors chemically. They are also in a dance with the concentrations of the nucleoside diphosphates and nucleoside monophosphates being used as energy conduits for reactions, the energy for the synthesis of DNA or RNA, or being used to attach a new phosphate, returning the supply of triphosphates. But those concentrations are in a reversible reaction with phosphoric acid and its conjugate bases and the hydrogen ions. And pH is determined by the concentration of hydrogen ions, and the pH affect the enzymes, and the proteins and nucleotide triphosphates are broken down into acids, and you can chalk it up to homeostasis, but feedback inhibition pathways. These macromolecules make up the chemical structure of a cell. Amino acids create proteins that catalyze reactions and form the cytoskeleton. Carbohydrates are used for energy in their bonds through the Krebs cycle, but ribose and deoxyribose are sugars that chemically bind phosphate and nitrogenous bases to form nucleic acids. The last is lipids. Lipids, or fats, waxes, oils, and hormones. Notice the phosphate, notice the tail and glycerol, notice the carbohydrate chains. Lipids form membranes for cells. Lipids orientate with their hydrophobic tails away from water, in other words, towards each other. They form in a sphere because this orientation is the lowest surface area needed and encapsulates concentrations of aqueous solutions, perhaps of nucleic acids, amino acids, carbohydrates, and some minerals. A cell, the nucleus, endoplasmic reticulum, mitochondria, have lipid membranes. Embedded in these membranes are proteins, enzymes. A virus may have a membrane of lipids or an envelope of proteins, encapsulating their genetic material such as mRNA embedded with the ACE receptor binding spike protein that will facilitate the delivery of the viral genetics into the cell. Once the mRNA is encapsulated by the cell, ribosomes will begin to synthesize the protein from the sequence in the foreign mRNA. A normal virus will create replicas in order to infect more cells. Injecting receptor binding protein embedded lipid encapsulated mRNA to access the ribosomes of cells will produce spike proteins so the immune cells can produce a protein known as an antibody with ribosomes in response to the spike protein concentration spike. Even if the mRNA sequence was an amino acid sequence of just spike protein, the concentration of amino acids, nucleic acids, will increase, causing changes in the equilibrium of hydrogen ions, causing changes in pH. Enzymes are affected in changes of pH or changes in hydrogen ion. Enzymes are also affected by the products of reactions called feedback inhibition pathways. Enzymes in the reactions and related enzymes in those reactions are called pathways. Products of a reaction in a chain of reactions can affect the efficiency of enzymes regulating the metabolism of a cell. Certain molecules, proteins, elements can affect the efficiency of enzymes called cofactors. So the concentration and changes in concentrations causes a dance of reaction rates, concentrations of reactants and products, or stoichiometry. Cells are chemically keeping tabs on concentrations and the cell's metabolism through feedback inhibition pathways, maintaining equilibrium. Even if a quote unquote good mRNA makes only exactly what was advertised through a sequence of the nucleoside phosphates, the mRNA and the protein sequence therein affect the concentrations of the cell, altering the equilibrium of the cell. The proteins synthesized from a foreign mRNA did not evolve with the cell, and proteins misfold. The foreign mRNA codes for an amino acid sequence that is foreign to the cell, meaning the natural proteins regulate through feedback inhibition pathways related to their function within the cell. The concentration is being checked, where the metabolism and catabolism of a foreign protein is an extraneous variable affecting the equilibrium of a cell. Maintaining equilibrium and constantly maintaining proteins, as well as the rest of the stoichiometry, costs energy. Trillions of reactions are occurring every trillion of a second, and the cell stoichiometry, feedback inhibition pathways, methylation of genes, etc., means proteins misfold. Proteins that evolve with the cell may have feedback inhibition pathways that facilitate its repair or recycle. However, misfolded protein accumulate as plaque. Liposomes created by the cell and proteins that break down proteins in the cytoplasm can deal with breaking down protein, but enough protein misfolding causes metabolic hindrance on the cells and affects the equilibrium of the cell. The metabolism and catabolism of concentrations of amino acids, nucleic acids, lipids, and carbohydrates thermodynamically tend towards entropy, disorder, lower energy states, so equilibrium is a constant fluctuation of concentrations and reaction rates, and costs energy. Metabolic hindrance leads to cancer and disease. A healthy cell will perform its function, but how does a cell perform its function? Proteins, filaments, and microtubules and marker proteins facilitate physically moving macromolecules. Natural mRNA may sequence a number of different proteins. So as membrane protein is being synthesized, so are related proteins. Foreign mRNA protein will be worked on by ribosomes, like a natural mRNA, but foreign engineered mRNA can code for a protein marker that delivers protein, RNA, and DNA to the nucleus. A lab-engineered virus and a mRNA injection perform the same job of delivering foreign genetic material to the ribosome. This is a map of interrelated reactions of the synthesis of lipids, amino acids, carbohydrates, nucleic acids. Glycolysis, lipogenesis, gluconeogenesis, amino acid synthesis, and nucleotide synthesis are regulating each other through their reactions. There is an enzyme present in snakes. that combines the precursors for the venom of snakes and secreted from gland cells. The bureaucracy of healthcare professionals receive kickbacks from the pharmaceutical and insurance industries. Doctors receive degrees, but that degree was approved by a bureaucracy of people who got degrees, approved by a bureaucracy that got degrees. So think neurons are cells that use ribosomes and mRNA that create the membrane protein, concentration pumps that maintain the concentration gradient of potassium and sodium that allows for the electrical discharge down the axon that signals the release of neurotransmitter, which are protein, maintaining equilibrium, why you would not inject mRNA, and how it's the same as a lab engineered virus. A Pandora's box of questions. There are trillions upon trillions of reactions happening every trillions of a second. mRNA, whether created by the cell or introduced through a virus or injection, will create a protein or spectrum of proteins that could potentially perform any kind of chemical or biological work, including creating snake venom in your own cells to creating a human with 4 arms if there were malicious intent. Regardless, a perfectly engineered mRNA would still upset the equilibrium of a cell, and though we are clever creatures, we should respect the situation we are in.